So the title of my talk that I wanted to present today is sort of a bit sensationalist, curing cancer with code. I guess the answer, spoiler alert, is no, or at least not yet, but we'll go on to talk about that. So I wanted to give you a quick introduction to who I am. So I was actually born in Brighton. I'm a Sussex girl. I moved away to do my bachelor's degree in biochemistry at the University of Manchester. Then I came back to Sussex to do my master's degree in molecular cell biology. And then I decided Sussex was too expensive, so I moved away again, did my PhD in chemistry. And this is where I first was exposed to code. My PhD was in computational chemistry and biophysics. So I taught myself Python and Bash just because I felt like I needed them. And I then ended up getting offered a job as a postdoc or a research fellow at Brighton Sussex Medical School, where I use a mixture of Python and Julia. So apologies, but there will be some Julia in this talk. So previously, I've done a lot of experimental work. My background is I'm a biologist, I'm a biochemist. My PhD was then very computational, and I want to be in the middle. So that's where I'm very lucky to be with my postdoc. I get to do some experiments and some computational work. So today I'm going to be talking mostly about some work that I recently have published on diffuse large B-cell lymphoma, which is a type of blood cancer. I wanted to share some more recent results at the end, but I've actually had a bug in my code for the last 2 weeks, so haven't been able to produce the results I wanted to, unfortunately, but there should be enough for us to talk about today. So the disease that I'm working on in my postdoc is diffuse large B-cell lymphoma. It's a very common type of blood cancer. And it's very interesting because it's an extremely heterogeneous disease. If you're not familiar with this word, it just means that even in between patients, they'll both walk into a clinic, they'll both get a diagnosis of DLBCL, is how I refer to it, but the disease and the mutations that make up their disease is very, very different. And there's lots of different subtypes where some patients will do much better and some patients will do much worse. So here is just an example of this, that you don't need to worry about the detail of this at all, other than the arrow along the bottom. Each column is a patient and each row corresponds to a mutation if they've got a little coloured in box. So you can see that there's lots of different ways we can cluster patients. So here they're clustered into clusters 1 to 5, but even within those clusters, there are big differences from one patient to the next. Despite this, everybody gets the same treatment. It's a one-size-fits-all approach, and it cures about 60% of patients. The rest have very poor prognosis, and around 1 in 3 DLBCL patients will not survive 5 years after diagnosis. So the current treatments that we have is an immunochemotherapy regime called R-CHOP. So we have these 3 chemotherapy drugs, this steroid and this antibody. And this hasn't actually changed for over 15 years, despite the fact that it's not great. I mean, don't get me wrong, it's not terrible. And in fact, adding the R to R-CHOP was revolutionary. It increased the survival from sort of 45 to less than 50% within over 5 years to about 60%, like I said. And lots of people will say that R-CHOP is pretty good, but I don't think 40% of people not surviving for 5 years is good enough at all. And again, I'm trying not to go too hard on the biology here, because I appreciate that this is an audience full of coding people, but I do need to explain this family of molecules that I'm working on. So I work on a protein called NF-kappa B, and it's part of our normal immune function. It's got roles in inflammation, but when it goes wrong, it does all these terrible things, such as causing metastasis, tumor cell proliferation, evading therapy. And this lines up really well with all of these cancer hallmarks. So NF-kappa B helps cells resist death, keep proliferating when they shouldn't be, evading things, invading and metastasizing, being immortal. So instead of dying when they stop receiving signals, they keep going forever and ever, and hijacking blood supplies. So this is a really important molecule. I say molecule, it's actually not one molecule. We know that NF-kappa B is important in diffuse large B-cell lymphoma, but there's actually a whole bunch of different types of NF-kappa B, and these form dimers, so two molecules bind to each other. So there's 15 potential combinations. We have some understanding about some of these subunits. So, for example, we know that CREL is important in some types of DLBCL, REL-B might be important in others, but overall, we don't really have much of an understanding. And one thing I like to remember is, even though when I'm doing my work in a Jupyter notebook and it's code on a screen, actually in the patients, this is all happening in a very complicated environment. It's not just cells sitting in a dish or code sitting in a code block. It's actually happening in the context of the so-called tumor microenvironment. So there'll be normal immune cells, healthy cells, interacting with the cancer cells, and the cancer cells will receive signals from these cells, and this complicates the picture further. So to kind of summarize, the complexity of DLBCL is patients have lots and lots of differences between them. NF-kappa B is very complicated, but very important, and we also have to consider the tumor microenvironment. We can't just knock out NF-kappa B with a drug, because this is toxic. As I mentioned earlier, it's really important in our normal immune function. We actually have it in every cell in our body. But if we could target the right NF-kappa B in a person who has, for example, too much REL-A, then maybe we can help that patient and have better outcomes. But NF-kappa B is very complicated, as everything in DLBCL is. So it's actually not just one signaling pathway, it's two signaling pathways, a canonical and a non-canonical arm, and these are kind of interconnected, and there's feedback loops. So NF-kappa B degrades its own inhibitors and all sorts of things. And even this simple cartoon representation that I have on the left, we can draw as a much more complicated computer model, which is on the right, where each of these arrows and each of these numbers next to an arrow corresponds to a reaction in the model, which has parameters assigned to it. And basically, we can give it a starting number, and it will do all the maths for us and spit out an answer. But you can start to see that this is far too complicated to understand without the help of computers, because there's so much interconnectivity, and it becomes impossible to predict how changing one parameter or one thing will affect other things without the help of computers. So this is why we need systems biology. So systems biology is, I like this kind of definition of systems biology from the Harvard Medical School website. It's using the tools of mathematics and computation because biology is too complicated for the unaided human brain. And I like to think of systems biology as kind of a cycle where systems biology is not just modeling on its own. It's very much tied to the biology. You have to identify the biological questions. You have to then get the data. You have to feed this into your model. You have to generate hypotheses or predictions, which you can then test, and then that generates more biological questions. I'm sure everyone is going to be much more familiar with computational models than I am, so I'll just gloss over this quite quickly. So the way that we make these models is we take some literature. So this is a paper by my boss where it's just summarizing some of the key systems in cell and in cancer. And then we do some maths. So we write these out, write every reaction out as an ordinary differential equation, which I'll talk about more in a second. And then we write some code to solve it. So, for example, if we take P52, this is a molecule that is chopped up by an enzyme or a protein called Nick. So it's P100, it's chopped up into P52. So we could actually write this out as P100 and Nick separately bind together to form a complex. So we have P100 bound to Nick, which is what this colon notation means. And then that turns into P52 and Nick. And each of these arrows has a rate associated with it. And the binding and unbinding of P100 and Nick, that's a reversible process. So we've got a K1 and a K-1, whereas something being chopped up, that's not a reversible process, so it's just a K2. And we can write an equation corresponding to that first section of that schematic, where we have P100 binding to Nick and unbinding. And we can use this to calculate the amount of P100 over time. And it's just a pretty simple equation where we have binding term and an unbinding term. So these equations are not very complicated. The complexity arises from the fact that there are hundreds and hundreds of them. I'm not going to really talk about this in the interest of time, but basically we can use these models to make some predictions, but we have to have some data to start with. And this was just a weather forecast from a day a while ago. So in order to get the data, this is my one of my very few experimental slides, we use this technique called flow cytometry. This is a really neat technique because you add tags to your cells, which bind to a protein that you're interested in. And these tags have fluorophores on them. So these are activated by lasers. And when you put the cells that have these tags on them through the flow cytometer, it puts them into a single stream of cells. So they feed through one by one, and the lasers shine and the tags fluoresce, and you can detect this. So you get a chart like this one in the bottom right corner, where you can see that there's a population that has high PE, which is one of the tags, but it has low APC, which is another one of the tags. And you can get information about different levels of proteins in this way. So typically, when you do flow cytometry analysis, you collect your data on a flow cytometer. You then export it as a so-called an FCS file. Typically, you'll use something called the Cytoflex software, which is what is the built-in software for the flow cytometer that I use, at least. It's not available on Mac, first problem. Then you select your data. So this is a process called gating, where you only pick the cells that you're interested in, because some of it's going to be rubbish. So you just make sure you're picking the ones you care about. The most commonly used program for this is called Flojo. And then you present your data with whatever plotting software you like. My problem with Flojo is that it costs money and it requires a license, which are not my favorite things. I'm very much in favor of open source. I think science should be for everyone and should be accessible to all. And I think proprietary software is very much against the way that works. It's also very powerful, but it can't do everything that I necessarily want it to do. So Simon is my boss. So I said to Simon, "Oh, well, I'm getting a bit annoyed having to borrow this Flojo dongle. Maybe we should consider buying a second one, even though I don't really want to spend money on it." And Simon said, "No, actually, you can code. Just use Python." So I found this Python module called Cytoflow, which is really excellent. And now I do all of my data processing. In fact, everything from after data collection, I do using Jupyter notebooks and Python. And my favorite plotting package is Seaborn, and this is my completely off-topic reminder that Seaborn colorblind palette should be used by everyone because 1 in 20 people are colorblind. So make sure you're using your colorblind friendly palettes, and Seaborn is pretty good for that. So here are some plots that I've made using my Python pipeline. And all of these are useful in different ways. So I've got some histograms. I can get these kind of neat 3-dimensional scatter plots showing when something's high in different markers. I can make bar plots. I can plot the fold change. But actually, it becomes quite complicated when you're trying to compare different things. When you're trying to compare lots of different proteins across lots of different cell lines. So we came up with this idea that we could collate all our data and standardize it to a Z distribution, which is a type of statistical distribution where the mean is 0 and the standard deviation is 1. And then we plot what we call N of kappa B fingerprints. So this is what they look like, again, plotted using my favorite Seaborn colorblind palette. Every DLBCL case has a different fingerprint. So this little orange guy down in the bottom left corner is healthy cells that I've taken from someone's blood. And then the dark orange guy is a patient sample. And then the other 4 are all DLBCL cell lines. So these are cells I've been growing. They originally came from a patient, but they're being maintained and they have been treated in such a way that they will live forever in a dish. And you can see that the healthy cells have very, very distinct, they're very homogeneous and in their expression of both REL-A and REL-B, whereas the cell lines have quite a wide representation of REL-B, and the patient sample has a bit of variation in its REL-A. So this is a really handy way for us to compare differences between samples whilst maintaining the within sample heterogeneity. This is one of my few Julia slides. I'll try not to plug Julia too much because I'm actually more of a Python person myself, but my boss is very, very keen on Julia. Basically, this is how we set up our models. So the reason that we use Julia is because for simulations such as these, it's much, much faster than Python. It's comparable to Fortran and C. And you can see in this little grid I've got in the bottom left that Julia likes to think it's very, very easy to learn, but it has the advantages of being as fast as Fortran and C. I would say it's not quite so easy as Python. I kind of think of Julia as if Python and MATLAB had a slightly ugly baby, and the error messages that you get with Julia are awful to debug, whereas Python error messages, you just type it into Stack Overflow and, oh yeah, second answer, there we go. Julia is not, it's a little bit more of a challenge. But basically you start with some parameter files. So these are going to say what numbers you should start with and what are the reactions that happen between those numbers. And then you'll generate this ODE model file, which is a list of all of those ODE equations I showed earlier. You then plug this into a solver. So we use a Julia module called differential equations.jl. And then you get a plot that looks like this, where you can see how each NF kappa B component changes over time. You can use multiple cores to solve lots of cells, and you can tweak each parameter set so it's a little bit different from its neighbor. So in a real experiment, not every cell in the dish is going to have the exact same number of proteins compared to its neighbor. There'll be some variation. So this is a way that we can kind of make it a bit more realistic. And you can do things like rapidly check the effect of changing every parameter. So if you want to get the idea about how a drug treatment might affect these cells, you could turn down a parameter and see what happens to the system. So we can use computers to do years of experiments in just hours. So back to my fingerprints. So on the left, I have my experiment, And then on the right, I've got some simulation data. So this is data generated from that model. So we kind of fed in some data from my experiments, and then this is what the model has spat out, so to speak, in terms of the rel A and the rel B. So those are the two NF-kappa B proteins. But we can go beyond this. We can predict how DLBCL cells respond to signals. So if you remember at the beginning, I said these cells aren't existing just in the context of cells in a dish. They're in this complex microenvironment. So this is some work done in collaboration with Aaron Pack, who's a PhD student I work with. And we looked at rel A bound to P50, which is one of the most important NF-kappa B complexes, at least we think, in DLBCL. And what we saw was that across these 4 different populations, the dark blue had the lowest, the dark green had the highest, and then the other 2 were in between. And when we stimulate them, so this is in response to a short stimulus in a simulation, which then triggers some changes, which then the changes fade away after the stimulus is removed. When we stimulate, we expect the blue one to actually be the biggest, even though it starts off without any stimulation as the smallest. And so here is an experiment where we can see actually it really nicely matches up with our model. So without any stimulation, the dark blue is the lowest, the green is the highest, and then the other 2, then yellow, then light blue. And the prediction, when we add the stimulus, the dark blue is the only one that really does anything. Actually, yes, that's what I also see in the lab, that when we add the stimulus, the dark blue is the only one that goes up and everything else sort of stays the same. So I appreciate this has not been super duper Python-y, but hopefully it's been interesting nonetheless. So the current treatments of DLBCL are not good enough, in our opinion. We know NF-kappa B is really important, but it's very difficult to understand the NF-kappa B signaling without computers to help. So we need systems biology. We've seen that every DLBCL patient or case has a different NF-kappa B fingerprint, and from the simulations, we can predict changes in NF-kappa B in response to stimuli. Tested these predictions in the lab, and I guess to kind of come back to when I said at the beginning, the title of Curing Cancer with Code, no, not yet. But the idea for this work is that we're working towards a future in which different patients could be treated with different NF-kappa B subunit specific drugs based on their fingerprints, and we could predict that using models. So kind of, I guess the take-home message, at least in my opinion, is in DLBCL, personalised medicine is the way forward, and computer modelling is a really important part of that. These are all of the people who have helped me with this work. So my boss, Dr. Simon Mitchell, is the tall guy in the middle, and then the Pepper team and Mitchell team as well. And Dr. John Jones is our clinical collaborator. So that's how I get my hands on chunks of people's neck that they've donated. And I'm funded by the UKRI. Yeah, thanks so much for listening.